So Aneesh came to us from Amherst College in Massachusetts. His project was focusing on water quality in Carlyle Lake, which is an important reservoir in this area. It's about 60 miles east of St. Louis. He was working with a team, Alex, who's going to talk tomorrow. We'll talk about the water quality part. But the other part, as Natalie said, Aneesh was the Managing Action Center for the Humanities Intern, so there was a social science part to his work. And one of the things he did was develop a survey and conduct that survey at Carlyle Lake on a weekend with Alex. So he's going to talk about those results as well. On a personal note, Aneesh I think is his first time in the Midwest, so it was a cultural experience for him to be in the Midwest and conducting a survey out at Carlyle Lake. So here's Aneesh.
Thank you, Dr. Sloan. My project is called Assessing the Role of Sediment Dynamics in Triggering Harmful Algal Blooms and Community Perceptions of These Blooms at Carlyle Lake, Illinois. So a little bit of background about Carlyle Lake. It's the largest man-made reservoir in Illinois, 41 square miles. It's part of the Kaskaskia River watershed, so the Kaskaskia River flows in from the bottom. It's used for flood control, and this is why water levels vary in the lake depending on how much water they let in via the dam. It's currently managed by the Army Corps of Engineers. And historically, the lake faces several problems, none of which warrant major concern, but these include nitrates, phosphates, all of which can cause algal blooms. So this is our hypothesis and objectives for the project. It's split into two parts. In the first part, we wanted to study the temporal and spatial dynamics of phosphorus, iron, and other species in the lake. We wanted to understand nutrient exchange along the sediment-water interface. And we also wanted to see how soluble reactive phosphorus, ammonia, and nitrate interact with each other. Now our second part, I'll get into later. It's about examining community perceptions about Carlyle Lake. And we wanted to find out people's concerns, how they engaged with the lake. So I'll get to that in a bit. So we wanted to collect sediment samples, and in order to do so, we needed to choose a variety of sites which would encompass the different conditions in the lake. Because as you can see, it's a pretty large lake, 41 square miles. So we had three types of sites, shallow sites, and you could see that there's a trestle that kind of bisects the lake right there. And beneath that, there's medium depth and deep sites. So now that we have our sites, this is how we collected the sediments. We used the Petit Ponar Sampler and some rope. So how this works is you would take that rope, put it into that loop there, and it's basically like a claw. So you would lower it into the lake, and it hits the sediment bottom. And as soon as you release the tension in the rope, it snaps shut, and you could pull the sediment up, put it into a Ziploc bag, freeze it, and deal with the sample later. So next up, now that we have our sediments, we could look at extractions. So this is a pretty complicated diagram here. But essentially it shows that there's fresh, wet samples, and there's dry samples. And on the fresh samples, we do a distilled deionized water extraction. So we check for ammonia, SRP, and nitrate in the pore water. And then we also do a 2 molar KCl extraction to find out the same stuff, but what's bound to the clay. And we put this on a DA. I'll talk about how this works later on. With the dry sample, what we do is, first of all, we take this, put it in the heating oven, and grind it via mortar and pestle. And then we could do a 1 molar KCl to find aluminum, a 0.1 molar barium chloride extraction from major cations. And we also do a total phosphorus via ignition and acid dissolution to find out total phosphorus. And a precursor to this step is finding total organic matter. And all of this metal stuff goes on the ICP. So this is basically the text of what I just talked about. There's basically two classes, pore water and clay bound. For all of the nutrient from the water, we put it on the DA. So how the DA works is, what you do is you put in a couple standards, it creates a calibration curve, and it measures absorbance in the samples. Now for the ICP, we use that for the metal samples, and what it does is it uses plasma to excite atoms, for those of you who don't know, and those emit specific wavelengths, and we can couple an atom to one wavelength because they have unique wavelengths. So this is the first part of my data. We see there's a moderate correlation between total phosphorus and SRP in the pore water. So this is water that's adjacent to the sediment, so it's bottom water. So we see there's a moderate correlation here. However, if you stratify and we look at only bottom sites, we see that the R-squared is tiny, even though there's a very tiny relationship between total phosphorus in the sediment and SRP in the lake water. For medium and surface sites, there was essentially no relationship because the R-squared was tiny. It was something we weren't quite expecting, but that's how the data came out to be. Now, this shows that ammonia and nitrate, they're inversely related. So as we get more ammonia at a deep site, there's less nitrate. And this is for all the deep sample sites where there isn't that much wave action, that much water. We see a pretty strong R-squared. So this led us to believe that there might be nitrification going on in the lake. So to confirm that, we did another graph. So this is dissolved oxygen at depth. So as we see that at the deep sites, as we get closer to the surface, dissolved oxygen increases. Yeah. And so this tells us that nitrification is taking place. For those of you that don't know, it's basically when ammonium is oxidized to nitrite, then to nitrate, and then there's bacteria that can convert the nitrate back to N2 nitrogen gas. So even though N2 gas is not dangerous, the precursors to that gas can cause algal blooms. So this was an interesting correlation we couldn't quite explain, but we do know that more sodium means more basic conditions, and this somehow correlated with more clay-bound phosphate. And there's a pretty strong correlation there. And this is probably the strongest correlations. It's copper versus aluminum in the DDI samples, and lithium versus aluminum in the DDI samples. So these two metals bind to similar ions, and it makes sense that you would see them occurring at the same time. So yeah, essentially we found out, for the first part of the project, we found out that nitrification is taking place, but only in the deep samples. We've also found out that water content stays the same. There were small fluctuations in nitrate and ammonia, but nothing that would change the findings. And we also found out that phosphorus is not impacted by iron, and it actually depends on organic matter decay. So for further investigations, we would like to study pH, because we don't have that data, pH and nutrient relationships, explore more about nitrification, and study long-term nutrient concentration. So chronologically look at how ammonia changes at a certain site, or something like that. Now our second part of the project was a survey, so people could tell us the answers and we could write it down, or they could write in the answers. So we talked about how they use the lake, potential concerns, how far have they traveled. So this is a photo of the survey right here. There's some quantitative questions, like data we could quantify, and some like subjective questions. So this is what we found. When we talked about perception of Carlyle Lake water quality, we found out that 54% thought the water was excellent or good. Interestingly enough, no one put down that the water was poor, and it makes sense because the water is fair/good. For public concerns, we see most people didn't even hear of algal blooms, so they put down a 1, and we have a few higher numbers here. So the average concern was 3.7 out of 10. Now, when we asked people about lake management, the vast majority of people wanted the Illinois Department of Natural Resources to manage the lake, followed by the EPA, the Army Corps, and City of Carlisle. Yeah, but a lot of people gravitated to this. So from the survey, we found out what people know about the lake, how far they traveled, and we noticed that a lot of people that lived by the lake, they didn't have much of concern for algal blooms, and a lot of them didn't even speak to us. So for further study, we wanted to see if there's some sort of R-squared between algal blooms and minutes traveled to Carlyle Lake, because we thought people who traveled further might be more knowledgeable about algal blooms. Because past 200 minutes, we see that no one put down a 1 for their concern about algal blooms. So this was something interesting we got from the data. And, yep. That's it, guys. Thank you. I'd like to thank NGRREC, everyone that helped me here, the Hasenmueller lab at SLU, and the Manny Jackson Center for the Humanities for funding my internship. Thank you.
Do we have any questions for Aneesh?
Did you think about reaching out to the farmers in the area that surround this lake to ask for their opinions?
We basically had, so we were actually by the beach. So we were by the beach, by Keysport, but we didn't specifically reach out to farmers. We did talk to a lot of people by the marina who were like loading their boat into the water. Yeah, I'm sure we might have talked to a couple of farmers because we got people from a variety of backgrounds, but we didn't ask them about their profession, so we wouldn't know.
Okay.
Yeah.
My question is more for just personal knowledge. How long can these blooms last? We actually had problems with them up at Hemp and Hopper this year at one of our study sites.
I'm not quite sure. I think they had left for quite a while. I think at Carlyle Lake, we did see something that looked like an algal bloom. And 2 weeks later, when we came to sample it again, it looked like it was still there. So I could say in like this lake, Carlyle Lake, 2 weeks. I'm sure it could last longer.
Oh yeah, ours at Hemp and Hopper, it was at least 3.5 weeks that it spanned. So I was just wondering. Thank you.
Okay.
Any other questions from the audience?
I was just going to mention something. I failed to mention that Dr. Elizabeth Hasenmueller at St. Louis University was Aneesh's co-advisor. So much of the work he did was at St. Louis University.
I'm wondering a little bit about your dissolved oxygen plot and whether you considered relationship with temperature and depth. So as you get closer to the surface, temperature's going to be warmer.
It is.
And how that might affect your use of that graph or that information for nitrification.
Yeah, so I think with temperature, we did create a plot, but the R-squared wasn't as high as we wanted it to. So we need to look more into that, yeah. Because we didn't get a strong correlation, I think.
All right, thanks.
Yep.
We have time for one more question. All right. Thank you, Aneesh.